are your regional sustainable development partnerships. We build access. We are an open door to help you connect with the University of Minnesota faculty, students, extension, and any other resources. So RSDP is hosting this forum today, and I just want to give you an anecdote about why. When I first started this job, and my husband actually put it in the Christmas card, it's been 20 years that I've been in this role at the regional partnerships. I guess it was Christmas card worthy. That says a lot because we had our first grandchild this year. When I first started this job, I went to a meeting out in the Lamberton area. And at that time, still lived in St. Paul. I'm also in the agronomy department. So I'm an adjunct faculty member in the agronomy department. And showed up at a regional partnership meeting. And there were two farmers there. One was an organic farmer, one was a conventional farmer. And at that time, they still did a lot of their financial book work on paper. And they sat at a table in a group, and they said, how do we make money? I'm an organic farmer. How do I make money? I'm a conventional farmer. This is how I make money. And they opened their books for the group to see, because there was some concern, like, are you guys crazy? Are you guys crazy? And so they sat together at a table and had that center court conversation about how we can farm in multiple ways and be successful in multiple ways. So that really imprinted on me that RSDP is a place where we can come together, have conversations, learn about the other sides. We can bring science, we bring community, we bring experience. There's different ways of knowing and approaching this work. And so I'm very proud that the RSDP is helping to host this conversation among farmers and seed dealers and scientists. So thank you again for being here. I think that was all I need to say for my opening comments. A big welcome. Thank you. We also have a number of people joining us online. So hopefully everybody who's online, thank you. Oh, there you are. Thank you. Welcome. We're very grateful to have you engaging with us online. This could be a long day to sit on 6 hours of meetings online, but we're glad you're here. And so let me introduce there's been a lot of work that's gone into this. We're very grateful. And Danielle Piraino has been our lead RSDP staff person working on this. Danielle works for our Southeast RSDP. She's our outreach specialist working with Andy Sutton, our Southeast Executive Director, who I'm guessing a lot of you are from Southeast. Any ideas, projects between now and Valentine's Day? Andy and Danielle, if you want to talk to, we can get something rocking and rolling. So Danielle has experience farming. Her background and degrees are in sustainable agriculture. She was with Extension Agriculture Natural Resources before we snatched her over to RSDP. So we're very happy to have her on staff, and she will be emceeing and kind of leading the conversation today. So I'm going to turn it over to Danielle. And thank you all again.
Thanks, Kathy. Thanks, everyone. I'm going to try and project. Can you hear me in the back? Yeah. Okay. If at any point you can't hear me, I will mic up myself, but it's a little feedbacky, so we're trying to avoid that. Yeah, so as Kathy said, I will be your host today, and I just wanted to thank a few people. So thank you to the Forum Advisory Group members and other U of M Extension staff, as well as our graduate research assistant, Maddie Hazy, who for the last several months have worked alongside me organizing this forum for everyone today. And thanks to our presenters for flying to Minnesota, some of you, in the middle of January. We appreciate it. So I just wanted to give a little bit more context and level setting to build on what Kathy already shared. And this will just be the surface, and we will get deeply into it today. But we know a few things. So we know that neonics are a tool in agriculture. And we also know that their non-target impacts cause question and some concern. And two types of neonics were designated as the surface water pesticide of concern by the regulatory body in Minnesota, the Minnesota Department of Agriculture, in 2020. We also know that everyone in this room comes from a different background, and some of you are farmers and researchers, anglers, hunters, or concerned community members, and we're glad that you're all here. And you likely arrived with your own opinion and perspective on neonicotinoids, I am sure. And so, like many agricultural topics, this can be sensitive and can be controversial, and we just want to name that up front. Disagreement and conversation is democratic so long as you do it respectfully. So our goal today is to share recent and relevant research so that you can make informed decisions for yourself, the land, and the ecosystems that you work within. We ask that you listen with curiosity and respect for our presenters and for one another. And many of the presenters today work closely with farmers or are farmers and understand these real world challenges of balancing pesticide, pest control, and environmental impacts. And so this is just a starting point for all of us to think together about how some of these non-target impacts may be something to pay attention to, and how neonics may fit into the long-term land management in our region. So, in order to make this time worthwhile for you and everyone here, we have some agreements for dialogue. You saw them when you walked in. We ask that you keep these in mind today as you're having discussions and when you're asking questions. So I will just quickly read them so we're all aligned. Today is about the research. So this forum is not about specific next steps, nor is it intended to promote a specific outcome. We ask that you assume good intent. We're all here because we want to do right by our communities in some way. Please respect different perspectives and try to understand where other people might be coming from. Most importantly, be open to learning. We have some really skilled experts here who are eager to share their, decades-long research with you in some cases. Keep questions as questions, not statements, so just to respect everyone's time. And expect loose ends and imperfection. So this is a big and sticky, complex topic. We're not resolving anything today, but we hope that you learn. This is how our day will go. So you're here now. The first segment of presentations will be an overview of neonicotinoids, how and why they're used, and some efficacy trial data will be shared. We'll have a 10-minute break after that. The second segment of presentations will be about detections and impacts of neonicotinoids on the Minnesota landscape and wildlife. We will have a lunch break. Tom, wherever you are, we will have a lunch break. He was like, I'm a hungry farmer. I don't see lunch on.
Sit out my class.
Yeah, fair. And then we will have a panel discussion after lunch featuring farmers and small family seed company reps to have an honest dialogue about what they're seeing with this topic. And we'll talk about supply chain challenges as well. Another break, and then it's our last segment, which will feature presentations from our colleagues at Cornell University who will be sharing some experiences that they have had. There's some upcoming regulation of neonicotinoids in New York State. And so they'll share with us what they've been experiencing and how they're supporting farmers through this transition. Speaker of the day, Dr. Shawn Conley from UW-Madison. Dr. Conley is a professor of agronomy and the Wisconsin State Soybean and Small Grain Specialist at the University of Wisconsin-Madison. Welcome, Shawn.
Thank you. Well, good morning, everyone. Again, my name is Shawn Conley. Y'all hear me fine? Good enough. All right. My name is Shawn Conley. I'm the State Soybean Specialist at UW-Madison. Today, I'm going to kind of walk through some of the work that we've been doing, not just in the state of Wisconsin, but more on a regional basis, looking at some of the work on neonicotinoids. So before I get started, I always like to inform farmers who funds my research. I always think it's important. So on a given year, roughly 80% of the research funding for my program comes through checkoff dollars. So that would either be the Wisconsin Soybean Marketing Board, the North Central Soybean Research Program, or the United Soybean Board. So almost all of my research is directly funded by and supported by farmers. So I just like to always put that out there so people know what perspective I'm coming from. And I also couldn't do this with a really good team here. So kind of my talk today is neonicotinoids, targeted tool, not tool coverage. And I think the main point is, and I know we're not supposed to make any grand explanations, but my point here is I'm not going to be advocating at all for banning neonicotinoids. That's not at all what I'm going to be talking about. Mine is how do we use this as a tool in our toolbox, if you will, for farmers. Okay. Now, one of the interesting things that we know on the corn and soybean platform is we look at roughly 94 million acres, I think, of corn were planted in the United States in 2025. Roughly 83 million acres of soybeans, all right? And then I just Googled this, so if I'm wrong, you can correct me, but I think there was 8.6 million acres of corn grown in Minnesota last year, and roughly 7 million acres of soybeans, okay? Of that, corn acres, about 99% of all corn acres has a neonicotinoid seed treatment on it. We're looking at that, we're looking at basically 94 million acres. So that's the corn side of things. The soybean side of things, we don't really know, okay? Our best guess is we're roughly looking at about 60 to 80% of seed is treated. Okay, we do know roughly that amount, we get that from the industry, but we don't really know what percentage of it has neonicotinoids. The reason for that, good, bad, or indifferent, is, if this will go for me, maybe not.
Here.
Oh, this way. Does it go now? You know, this is always happens to me when I come to Minnesota. Maybe like there's a cheap ad about it. It's not, I just go with 10% rule. Not 10 smarter than 10% of what you're working with, it's not going to work.
It doesn't work 100% of the time, 10%.
And the other thing is, I was just making this comment. You know, after we were humiliating defeat by the Bears last weekend, I know how it has to be a Viking fan now. So, I didn't throw that in there. Okay, so back to what I was trying to say. Is the reason why we really don't know about these acres is this thing called the treated article exception. That this is kind of what do we call a loophole? Some people call it a loophole. Other people will just say it's a way to be able to not have to give every single seed treatment combination a specific go through FIFRA label. So I can see both sides of this thing, but that's the reason we don't know. You look through the AIs, so you have to report it on pounds per year usage, but we don't know how much was on seed treatments. Does that make sense, everybody? It's kind of you don't really know. So that's why we're sitting at on the soybean side of things. Okay, so why and when are neonicotinoids used? I know some of the entomologists here in Minnesota will probably do a better job than an agronomist talking about this. But I've seen there's an extensive list of pests that they use a label for. They actually do a very good job for the most part of being efficacious. But from an agronomist perspective, so I'll leave that to the entomologist to talk. From an agronomist perspective, where do these fit into systems? Manure systems. What are we known for in the state of Wisconsin? So that's green bay factors. Dairy cows, lots of manure. Okay, so we have manure systems. These are where these can work into. However, you also have to understand which neonicotinoid you use for seed corn maggot. So we usually, and I'll get into a problem I'll talk about later, is what's on your seed. I'll kind of give you a preemptive strike if that's going to be my last slide for you farmers in the audience and those online, what's on your seed. Because you may know there's a seed treatment on there, but if I ask most farmers, they do not know what the active ingredients are in that seed treatment. All right, go back to this. What are these usually targeted for? Manure systems, transitional systems. We will probably see this in another 5 years with corn and soybean price being what it is. We'll probably be seeing corn and soybean acres getting to pasture, going into some kind of conservation systems. And then when we get $8 corn again, we'll see the corn and soybean acres move back at that's into production. Those are places where we can see these seed treatments fit into these places because of the insect populations. Continuous cropping, we see a ton of that. We do in parts of Wisconsin because of our corn silage system. We see a lot of corn on corn on corn. We see some buildup of pests. We add manure to the system. Another good fit. And cover crops. So in theory, cover crops are where you would think this would fit, but we'll come back to that in a second. All right. Let's talk about some of the things here. Well, let's talk about cover crops right now. I can't remember where we stuck this one in here. So one of the challenges with cover crops is we see typically an increase in slugs. All right, we've seen this in the state of Wisconsin. Now, one of the challenges when we have cover crops, we see an increase in slugs. We also see, neonicotinoids were initially recommended in these systems. However, we see those impacting some of the predators on slugs. We actually see an increase of slugs in these cover crop systems. So in a sense, we're like pulling these out. These are actually exacerbating the situation. We're seeing more slug damage in cover crops when we have neonicotinoids in that system. All right. So from a farmer perspective, this whole thing with physiological responses is actually down. I'll talk about a research paper we published in a little bit about small plot stuff and how I get beat up all the time by industry for the small plot stuff. But as part of the North Central Soybean Research Program, we did surveys across the entire North Central region, which is roughly 82 to 85% of U.S. soybean production. So in this time frame, we were able to capture data on over 6,000 farm fields and over 600,000 acres of data. So we've seen data from actual farm fields, survey data, big plot information, looking at how some of these interactions occur. And one of the things we noticed in here was that we really want farmers to plant early. Planting early is a big driver in predicting soybean yield. All right. It's also when most farmers would think about using a neonicotinoid seed treatment, right? You're planting early, you're going into a stressed environment. We'll get to that in a hot second here. Now, one of the things we did notice is a few years ago, we did some work with thiamethoxam and cruiser. How many of you have ever heard of the bigger effect? I don't even know if they talk about that anymore. But with thiamethoxam specifically, we did see a physiological response to soybeans in our early plant situations. By that, what I mean, we're actually able to measure that the plants grew faster, more leaf area. So if you had a side-by-side, if you're a farmer, you've seen these, treated beans versus non-treated beans, the cruiser planted, the thiamethoxam treated beans look bigger, healthier. And we were actually able to capture that this information. A lot of that was more of a physiological response than it was necessarily an insecticidal response. Does that make sense, everybody? Because it was kind of triggering that. Because we did not see that in the metacloprid, clothianide. Typically, this was something unique to thiamethoxam. And for you, so to be clear, thiamethoxam is cruiser. Okay, just so we kind of know. Metacloprid is generic. It's pretty much in a lot of other seed treatment. All right, now back to this whole thing. So I'm from Wisconsin. I started doing this work. I've been in Wisconsin 18 years. I started working on neonicotinoids soon after I got to Wisconsin. And I was initially one of the biggest proponents of using neonicotinoids in soybeans. If you look at some of my early papers, I would probably the poster child of neonicotinoids. Are great, okay? However, as a scientist, part of our role is to adapt. You collect more data. You're informed more. You maybe change your mind. In politics, we call that a flip-flop. Okay? I don't think we hopefully call that in academia a flip-flopper. It's just being informed by new data to be able to change your mind. So around 2018, I started hearing reports from my friends like Christian Krupke in Indiana and other entomologists across the country. Like, this stuff we're not seeing the yield response. So we were able to collect data from 194 trials across, you see all these different regions. So we had about 11,000 plots from across the entire Midwest country. And this is the yield response. A lot of funny numbers, but in fact, did we see a yield response in neonicotinoid seed treatment? Yeah, we saw about a half a bushel. Okay. So on average, that's what we saw, half a bushel. So if you think, what's the average cost of a neonic? Probably generic, it's $2 a unit. Some of the higher ones is $8. What's the price of beans? Right now, $8. And depends how good job you did pricing, bargaining. If you're North Dakota, we're not going to talk about what the price of beans are. Okay. But the point here is that we're really not seeing that return on investment at this level of response. And again, we looked at this in different populations, fungicide, insecticide, different seed costs, different commodity prices. And in short, at best, farmers just basically broke even. So in terms of the profitability of this system, it's probably just break even at best. Again, talking averaging across all these different plots and fields. How much time do I got left? Just so I know.
Oh, you're doing great. 15
Perfect.
minutes.
Doing
Yep.
great.
You're doing so good.
She said you're doing
Can
good enough.
we, like, you, sorry, I'm just gonna.
I'm just wandering too much.
No, no.
Online
We're gonna mic
audience
you for
is
online.
So I couldn't sleep. I got up at 4:50 this morning. I started drinking coffee. It's not a good thing. You know, I'm usually jacked up as it is. And it was funny because I pulled into Quick Trip because who does not love Quick Trip?
Yeah.
Okay. I pulled into Quick Trip. Yeah. And I opened up the door and Metallica is jamming out of my car too. So you know I was getting amped for this talk.
Can you hear me?
Yeah.
This is not the best.
We need to stand back there.
That helps if you.
All right, I just stand back.
Can you stand still?
No. All right.
You don't have to wear it, but if you hold it a certain way, it won't pick your stuff. So you can wear it.
Is this better for everyone online if I?
This is the online speakers.
I need to ask, so.
I think just use them.
Just use
if you
this.
can.
All right. Sorry about that. This is me. All the Wisconsinites are used to me by now. You all don't know who I am, or this guy is crazy. All right. So now we're going back into this whole idea, you know, on what soybeans and neonicotinoids. Now, one of the things we promote is basically we tell farmers to buy a bag an acre, 140,000 seed count. Okay. Now, one of the things we also notice with, you know, neonicotinoids and fungicide seed treatments as well is we will generally see a reduction in stand when, or an increase in stand, usually about 10%, you know, 10 to 15%, depending on the environment. So if you take 140,000 seeds and 90% germ and you add a 10% reduction in germ, you're sitting around about 100,000 seeds, live seeds that are going to be coming up. Right? Our threshold for replants is basically 60,000 seeds per acre, or plants per acre. So what that tells me is that even in these early planted situations, we can lose half a stand of soybeans and still come up with roughly 95% of yield. Okay. In these early planted situations. So I just want to kind of talk about this in terms of some of the risks associated with it. Now I'm going to credit Christian Krupke. So this is new data. This hasn't been published yet. He's in the works of being published. And what he did is he actually went in and looked at, because it hadn't been done yet, it's the academia, I guess the industry probably has some information to this, about how much seed treatment is being expressed in different plant parts. So if you look at the label on soybeans, and if anyone here is from Syngenta, they can correct me if I'm wrong, but I think it is going to be like 0.0756 milligrams per AI per gram per seed. So this much, okay, not a lot, okay? What he did is he went through the different growth stages and figured out how much of that total amount of AI that was put on each seed is expressed in this different plant parts. So here it is basically pretty slug, I guess you call it slugs pretty than that. So this would be the EE growth stage. Look at here, of that total amount on a per seed basis, 14.6% is expressed in cotyledons. 2.5 is in the hypocotyl, about 1.8% is leaves. We get out here to V1. Again, you still see most of that neonicotinoid is sitting where those cotyledons. You look at the first true leaf, where we see some of this activity from slugs in the foliage, how much neonicotinoid is sitting there? Not a lot. So the question is then, again, that's a total percentage. If we subtract this number, 100. So who's good at math? 0.5%, 0.7%, 12.7, 1, 13, 14. So over 20% of that amount of neonic that was treated that seed is actually in the plant. Okay, where's the other 80%? So, exactly. This is cool stuff. It's never been done. The company does a really good job, and you should have had him come here and sit with me. So he would have said that. All right. Now, remember that big survey I talked about, 8,000 farm fields? We pulled this data about planting day. We also looked at how predictive seed treatment is. Where do you see seed treatment on that list for predicting soybean yield? Pretty darn low. Not really a predictor at all. Okay. And we looked at this, we go into all of these farm fields, and we see on average, and you all heard of Group 1 beans in Minnesota, on average, we're looking at, this is farmer data, right? Probably 100,000 acres from Group 1 to 30 Group 1 region here, 23 bushels per acre, is what farmers were getting on their farm associated with neonicotinoid seed treatment. They're able to pull this off. So this is kind of a sense, gives you a sense of what we're seeing on the soybeans. All right, last thing, risk. Why do farmers do this? Farmers are not, I come from a dairy farm. I hate cows, so I don't want to milk cows. Not gonna lie. I've been milking cows since I'm 18 years old. Love soybeans. Never been kicked by a soybean. Swatted in the face by a dirty tail by a soybean plant. But why do farmers do this kind of stuff? Okay, why do they add these inputs? They want to minimize risk. Most farmers are pretty risk adverse. They run a lot of acres. They don't have a lot of work. They have a lot of work. They don't have a lot of workers. It's really hard to get across all their acres and manage all these acres. So they're looking at applying inputs to minimize risk. So just for fun, we went through and looked at these intensive systems and what farmers do to minimize risk. And does it actually minimize risk, or is this something that is sold to a farmer that they think they're minimizing the risk? So what we did is we went through and did some of this work. Seth Nave did some of this in Minnesota here, probably at Waseca. I think Waseca is probably on this map right now. So we got some data from here. And we looked at these high input, we call them kitchen sink trials. We put everything except the kitchen sink into these trials. Like insecticide, treatments, biologicals, foliar feed, you name it. And then we basically did pretty much just kill your weeds, keep your soil fertility level good, plant good, varieties, basic management systems. And what we found from this, here's the state of Minnesota, bottom right, second over from Wisconsin, is that in fact, these high input systems don't even come up to average for minimizing risk. Do they increase yield? Yeah, they increase yield. When we have more inputs, what is those input costs? Higher. So you're actually not minimizing grower risk by throwing all these kitchen sink products at your farming system. All right, just want to make that point. And this is where I kind of want to end this. Like what's on your? This is really important, super important. Is that there's a lot of seed treatment products out there. I really want to encourage farmers to dig in underneath, the toolbox, if you will, and actually know what you're putting out there for seed treatment. There's a lot of things that are applied, but we really don't know. We update this every year. David Smith, our pathologist, does this. But I told you that I planted Nitro Shield 4 as a seed treatment. Okay, what the hell is a Nitro Shield 4? Anybody? Anybody? Well, Matt Holbert is in there, so you have a neonicotinoid. Okay. How about kind of poncho? Seed treatment. What poncho is. But Senator 600 FS. You don't know what's in there. My point is when you talk to your seed company, understand what is on that seed so you have a better, clearer idea of what pests you are managing. Because again, something like a Matt Holbert maybe not doing the best job on seed coordinating, correct? Right. Well, there's things we have to I guess the last thing, how much time have we got?
We have 10 minutes left.
What?
But
Yeah, you started early.
Yeah, started early.
You started early. You can end early. No.
You never tell me that. I guess the last thing I want to kind of make a point to is just going back to the risk side of farmers, and I'll kind of end with this one so I can leave more time for the next speakers, and we'll be late. We don't want to be late for lunch, right? So, biggest point that I always like to make is it's really hard. It's almost impossible on the corn side to get non-neonicotinoid treated seed. I'd say it's like, unless you call like 2 years ahead of time, or you're an organic farmer, it's really hard to get access to that. I understand seed companies will be talking later on this afternoon about pipelines. There's logistics. I get that side of the industry. Okay, I get that side of things. But that's usually plant seed treatments on corn are usually done at seed company place. Like soybeans, however, that's usually treated downstream. Like your local co-op does that treating. That's where you can make some decisions when you make these seed treatment choices. Challenge is for a lot of these companies, seed companies, if you want a free replant, is what our crazy agronomists like me telling farmers to do, plant early, okay? And as part of Minnesota, probably the insurance date for planting early is April 15th. Am I correct, crop insurance people? I think it's probably April 15th here. We're gonna call it April 15th, 20. But to get insured by your seed company, you need to typically have a full seed treatment load on your seed in order to get a replant for that seed. So I'm telling farmers, plant early. It's free yield. You're going to gain a half a bushel per acre per day by planting early. You go tell your seeds person that, well, if you want free replant, just in case there is a whatever, you get a bad stand. Well, I'm not going to cover your seed if you don't put a full seed treatment load on. So that's kind of the rub, okay, out in the industry side. So with that, I will stop. I talk too much. Apologize. I'll be here all day. What else do I have to do? Listen to everyone else. Thank you for your time. I appreciate it, and see you later. Go Packers.
Yeah. Thank you, Shawn. Shawn will be available for questions at the end of this segment. So if you have any, which I'm sure you do, write them down, please, so you don't forget. Next, I would like to welcome Hunter Ness. Hunter is a researcher and master's student at the U of M, working in the game corn entomology lab and the cooked soybean entomology lab.
Yeah, thanks everyone for having me and for being here. I'm going to be presenting on some of our research we did at the U of M on early season soil insect pests and some efficacy trials for insecticide seed treatments in corn and soybean. And I'm probably going to overlap a little bit with Shawn and our next presenter a little bit, so I'll try to juggle that around. But insecticide seed treatments or ISTs, as Shawn said, 2012 to 2014, 90% of corn and about 76% of soybean, but probably 99% of corn these days. And we can see these graphs. You can see the rise in usage from 1995 to 2010 in use of ISTs, and this data is from North Dakota. What pests do they target? We have a couple popular brands here and pulled up their labels. So we can see both treat for white grubs and wireworms, which are early season soil insect pests. And for soybean, they also provide an early season protection of seedlings against injury by aphids. And at higher concentrations, or the highest concentration sold, they do provide some early season protection from corn rootworms. These graphs from USGS show estimated use in these three neonicotinoids each year from 92 to 2019. The units are in thousands of kilograms. And in 2015, they stopped including ISTs in their neonicotinoid estimates. And you can see where they stopped doing that, basically completely dropped off, which shows us how much ISTs are putting neonicotinoids in. They're basically the primary driver of neonicotinoids into our fields. So in part, due to the widespread occurrence of these neonicotinoids in all of our fields, there are some emerging concerns. We're starting to see some contamination in groundwater, in corn and soybean producing regions. And there's also concerns over the effects on pollinator species such as honeybees and monarch butterflies. So we want to better understand just the pest pressure in these fields and to understand the necessity of IST in these fields. And we want to better understand IST efficacy in corn and soybean pests such as aphids and corn rootworms. So I'll split this up into three parts. We'll talk about our early season soil insect pest survey, then we'll go into my soybean IST trials, and then the corn IST BT trials. So our early season soil insect pests that we focused on were wireworms and white grubs. Both are larvae of beetles that feed on seeds and find roots of seedlings. And they're difficult to monitor because these species can live up to 11 years in the field as larvae, certain wireworm species. They're persistent in fields, unlike seed corn maggots, which are kind of more ephemeral. And they're very difficult to monitor because they also migrate deeper in the ground in the summer, and the plant injury is usually not visible until it's too late. Since these are some of the primary targets of ISTs, we would like to know what the current pest pressure in Minnesota is like. So we published a crop news article looking for farmers who would be interested in participating in the survey. Asked for field characteristics in this survey for higher risk fields, high organic matter, history of seed treatments in the past, no-till, and ended up sampling from 52 fields all over southern Minnesota, as you can see in the graph here, about 25 each year. We use a couple different sampling methods to find these. We used soil inspections, which just consisted of digging out 5 cubic feet across the whole field randomly and hand sifting for these insects. And then we also deployed some baits, which consisted of germinated wheat and corn buried 6 inches underground. And we would come back for those 12 to 18 days later, bring them back to the lab and look through those for wireworms, white grubs, or any other insect we happen to find. And the economic threshold for both of these sampling methods is 1 insect per subsample. So that would be 1 insect per soil inspection or 1 per bait station in each field. So onto our results. This shows just the pest presence, whether we found anything or not, and kind of a map, so you can sort of see it's pretty generally distributed. There's not really seeing any major patterns here. And in 55% of fields, we found 0 pests in any of our samples. In 37% of fields, we found low numbers between 0.1 and 0.2 per subsample, which would be about 1 to 2 pests across all samples in the field. And then the remaining 8% of fields were between 0.3 and 0.7%, 0.7 pests per subsample, meaning we never reached economic threshold in any of the fields. So we can maybe conclude that this early season soil pest pressure is low. So this was design modeled after another study done by Dr. Smith in Ontario, and they found very similar findings. They found wireworm and white grub presence detected in over 50% of fields, but their presence and abundance were not related to field class. All right, so we have an idea for our early season soil insect pest pressure. Now we'll talk about our soybean IST trials. Soybean is attacked by a lot of different pests, including wireworms and white grubs in the early season, but the major pest in Minnesota is the soybean aphid. Neonicotinoid treatments are labeled to protect against soybean aphid, as we saw, at least in the early season, but we wanted to look at some alternative treatments. So we also are looking at diamide insecticides. These were introduced in the late 2000s. The first diamide pesticides targeted lepidopteran pests with a novel mode of action that had lower non-target effects than neonicotinoids. And then cyantraniliprole was developed in the early 2010s and targets various target pests with reports of efficacy against hemipterans, but notably it's not labeled for protection against soybean aphid. But we wanted to do some research and see if it had any effect on our soybeans. So our purpose was just to better understand ISTs in soybeans, and we looked at stand counts, above ground insect counts, and yield. This was conducted in 2024 and 25 in Rosemount and Lamberton at the research stations. We used a randomized complete block design, setting up 4 blocks of 5 plots, each 4 rows wide and 9 meters in length. And we planted each plot with one of 5 different treatments listed here. We have our untreated trial, which everything is treated with a base fungicide, I should say. So the untreated is just the fungicide. Then we have our 2 neonicotinoid treatments at a low and high concentration. It's in Cruiser and thiamethoxam. And then we're doing our diamides, using Fortenza brand of cyanotraniliprole at a low and high concentration. For insect pressure, we conducted foliar sampling, checking the leaves and stems from 10 plants randomly sampled from each plot. For data analysis, we calculated cumulative aphid days, or CAD, per plot, and this is just a metric to measure pest pressure throughout the season. The end of the season, and now to our results. So looking at our insect injury or aphid pressure, CAD, we can see that the high concentration of thiamethoxam did show significantly lower cumulative aphid days than our untreated trial, as well as our both of our diamide treatments. So that was significantly lower. However, we found no significant differences between stain count or yield between any of the treatments. Why is this? Well, you can maybe guess one of the reasons is ISTs are an early season protection. Aphids are a more mid to late season pest, with their numbers really peaking kind of later in July. So the ISTs just simply aren't don't have the efficacy while the aphids are at their peak. And these results aren't too surprising, as there have been some other papers that Shawn brought up before previously about showing that ISTs provide maybe negligible benefits to soybean farmers. All right, so now on to our IST trials in corn. Now we'll be focusing on the corn rootworm. This is the most significant soil pest in corn in the US and in Minnesota, definitely. There is an estimate that they cost about $1 billion a year in yield loss, and cost of control. Crop rotation was historically the main control method for corn rootworm, corn soybean crop rotation. However, corn rootworms are extremely adaptable, and they have adapted to crop rotation. Western corn rootworm, there is a variant that is feeding an egg-laying soybean. You can see from this figure here, you can see the range expansion of the rotation-resistant Western corn rootworm, and I believe some are found in southeastern Minnesota. And meanwhile, the northern corn rootworm went a different route, and they have extended their diapods to just wait out the soybean rotation and stay as eggs for an extra year, and then pop back up when you go back to corn. So they're just very adaptable, very sort of devious insects. The current primary management strategy in corn is BT technology, which was commercialized for corn rootworm in 2003. And as of last year, accounted for around 86% of planted corn in the US. However, rootworm again began developing resistance to BT. We can see throughout the years the different BT proteins that target rootworm. They became resistant in 2011 to Cry381, Cry34, 3581 in 2013, 2014 and 2016, a couple more, and then in 2019 there was resistance to 2 traits together, pyramided traits. So a number of strategies have been implemented to slow this resistance. Seed companies are selling corn seed with pyramided traits, as we said, to hopefully slow any resistance to one specific trait. And refuge is being recommended and sold in the bag as well to maintain susceptible populations in the field. And we want to look at some other methods as this resistance grows, including ISTs. So this purpose of the study is to look at the interactions between ISTs and BT in the management of corn rootworm. This was also conducted same places, same times at Rosemount and Pemberton in 2024 and 2025. Used a randomized complete block design with 4 blocks again, and each plot is 4 rows of corn and 9 meters long. We wanted to test the highest recommended concentrations of ISTs with current BT technology. So at the top, we used BT Delapro as our sort of non-BT control that does not target any below ground pests. It's just for European corn borer and other above ground pests. And then for our targeting, VT, we use VT4 Pro, SmartStax Pro. We tested those against 4 different IST regimens. We have an untreated regimen, and then we have, and then we also look at thiamethoxam at its highest concentration, clothianidin, and chlorantraniliprole, which is the diamide insecticide. So it gives us a total of 16 treatments. We checked for root injury in mid-July. This was done by sampling 5 plants randomly from the middle 2 plots of each plot, washing the roots, and then examining them from damage and measuring them, rating them with this Iowa State University scale. It goes from 0 to 3, with 3 as being a higher scores meaning greater root injury. And we calculated percentage of lodge plants, mean root rating per treatment, and then mean yield per treatment, and pooled the data from both years and locations. So onto our results. On the x-axis, we have our 4 different BT hybrids, and then the 4 different colors are our different IST treatments, which you can see listed in the top in our legend. For percent lodging, while we saw some differences between our BT plants and non-BT, we saw no significant differences within our non-BT plants. Looking at insect injury, again, we see some differences. The BT plants seem to protect pretty well, which shows that the corn are susceptible to BT in these fields. However, we didn't see any significant differences between the insecticide seed treatments in our non-BT plants. And then looking at yield, we actually see less significant difference between the BT and non-BT plants, but we did see a numeric difference. See, I highlighted there. Numerically higher yields in our non-BT group with the thiamethoxam, both of our neonicotinoid treatments compared to our untreated version, but it was not a significant, they were not significantly higher. So to summarize everything, I know it's early still, but summarize everything. Our survey showed that early season soil insect pest pressure was low in these fields. We saw in soybean that IST can reduce pest pressure, but we did not see any benefits to stand count or yield. And with corn, we saw lower lodging injury in the BT corn, but with yields, we only saw some numeric differences in yields between the ISTs. So some takeaways from this. IST usage remains high, and it is primarily used prophylactically without going out, monitoring, collecting data, seeing what's out there to see if it needs to be used. Early season soil insect pest pressure appears to be low in Minnesota and Ontario, at least, where so far we've conducted these studies. ISTs have limited effectivity against major corn and soybean pests. As we saw in our trials, while they prevented some injury, they provided limited yield benefits. And ISTs provided no additional benefit to BT corn in susceptible corn rootworm populations. Future work that we could continue to do is better understand these IST target pests, presence, abundance, and interactions with IPM strategies. I just think it's still kind of this big black box of wireworm, white grub, all these insects that are on the label for these pests, how much are actually out there in these fields. IST impacts on natural predators would be interesting to see. I know some of our diamide treatments had some kind of numerically lower numbers and yields and higher insect injury, which maybe there's something there with they're taking out natural predators. We don't know. We'd like to look into that more. Determine extent of IST usage in agriculture. It would just be nice to know how much we're using, but as we know, that's not really possible at this point, but there's some way we could get numbers on that that would make my research a lot easier. And the effects of ISTs on BT-resistant corn rootworms. Since we saw clearly that the corn rootworms in these trials were susceptible to BT, it'd be interesting to see some of those BT-resistant corn rootworms in effect with ISTs. And then, yeah, I'd just really like to thank my advisors, Bob Cook and Faye Yang, as well as both the labs and Minnesota Department of Agriculture, University of Minnesota. And yeah, that's all I got. Thank you all so much.
Next up is my extension colleague, Dr. Anthony Hanson, who is an entomologist and regional educator in integrated management, or IPM, for field crops. He's also the host of the U of M IPM podcast, which you should check out. Anthony also is a farmer and a beekeeper.
All right, yeah. So thanks for the introduction. I think part of that reason for including that is kind of what perspectives we're coming from. And I guess I kind of trained as an entomologist, working in integrated pest management. I'm the co-coordinator for Minnesota's IPM grant program. So I kind of get to work with all the different pest management areas, but also still being involved in the family farm, being more of a hobby beekeeper, but doing it for 10 years. I kind of get to dabble in all the topics that touch on this a little bit here. So what I want to focus on for this one is more so in the extension world, kind of what do we do a little bit? We're going to move away from the research side just a little bit and what's happening on the ground. What do we do when we take that research to the farmers, try to figure out what's going to work best in their farm? Then also on the extension side, we're hearing back from the farmers and what problems are they facing? What's going to work best for them for whatever tactics they can use. All right. So I just want to start off, what in the world's integrated pest management? Sometimes it sounds like a little bit of a jargony term, referred to as just IPM for an acronym. But it's really something that boils down to how do we minimize risks from, in our case, farming or pest management strategies. That's one risk for the farmers. We have our economic risk there. We don't want to be losing yield. But we also don't want to be losing money to things we don't need to spend on. That we talk about that a lot in the fertilizer world, but also pest management side. There's some things we don't need to put out there. That's kind of the hint here. How do we manage that question with neonicotinoids and other seed treatments? There are different ways to navigate through that a little bit. Some are pretty nuanced. Some are like, yeah, we're not seeing clear benefits. But I have this chart up here partly because this shows all the different tactics that do get used. And up at the oh, there's a little laser's not showing too much, but up at the top, it's going to have pesticides. That's one people think about a lot, but we have a whole lot more in the toolbox. We got our natural enemies or biological control, host plant resistance. That's your plant genetics, whether it's traditional breeding or BT traits on your transgenic side of things. And a few other things like your crop rotation, especially in the insect world. We talk about interfering, maybe you have pheromone traps that basically interfere with the insect, either disrupt mating, things like that. So there's a whole bunch of things in that toolbox we put together and figure out what works best for the farmer. And that's not just us deciding an extension, but it's the farmers themselves. So I'm kind of speaking from wearing multiple hats here. If I'm wearing my farm hat, I'm thinking about, okay, what's going to work in this toolbox? What specifically is going to work for one field may not work for another. And I have to run through all those different decisions. But when we talk about those risks, it's not just for the farmers. That's the financial side, but we're also looking at human health, the environment. So obviously pesticides are going to come up a lot for that. So we're really balancing all that and trying to figure out how do we reduce the risk for all of those. All right. And I always like to mention, farms aren't just field crops. I'm just going to mention like just an example on our farm. We have beef cattle. I do have honeybees, kind of the hobby side a little bit there, but if you have apple trees or other things, in this case, I have some of our, pretty healthy bumblebee populations we have, at least around our area, at least. That's kind of where we talk about environment a little bit more, what else is on the farm. So these are all things, you want to be doing well if you have that going on in your place, whether it's part of your farm operation, kind of a little more of the hobby or on the side kind of things. So this is one where I just like to stress that, at least I'm one good example where I kind of have interest in all of these here. It's not just one area. All right, so kind of moving to what we do a little bit for recommendations. First, I want to mention, I believe the Department of Agriculture on the state side can be here. They do have recommendations called best management practices for neonicotinoids. I think your packets should have a couple links in there, so you could look that up yourself or just Google MDA best management practices for neonicotinoids. Just kind of want to talk a little bit about navigating those. Kind of just key highlights. It's pretty much things that make sense, but there's some things that, folks may not be familiar with too much. First off, we're using IPM, integrated pest management. We're trying to save those seed treatments for the high risk scenarios. So before you heard about a lot of these cases where kind of at a high level, a lot of these pests, especially our major pests, there's maybe not great return on investment for using those neonic seed treatments. But there are a few others who talk about like, wireworm situations, a few others I'll mention later, where what about those farmers who do end up with high numbers? They're harder to find. And I know we have Bob Cook, our soybean entomologist in the audience here too. I remember at some research trials where we're trying to find cases even years before Hunter was doing research, and it's just hard to find high infestation fields, even if you set up the right conditions. Following the label, folks weren't aware of that. Actually, following pesticide labels is federal law. So if you ever go to pesticide trainings where we have to recertify folks, that's something we always mention. But also folks in kind of maybe going to, let's say, Menards or Fleet Farm, any yard and garden store, picking up herbicide, it's the same case there too. So that's something we kind of try to remind folks about, because there's a lot in those labels, either you're required to follow, or there's a lot of recommendations that are going to reduce impacts from those, especially for these seed treatments here. Reducing dust and drift, that one hasn't been mentioned yet, because a lot of times the biggest risk for these seed treatments, if you look at that picture, honeybees, it varies how resilient they are to some of these, but a lot of times those wild pollinators like bumblebees are more susceptible, especially to dust coming off planters. So you might have some planters that are more pneumatic. They're basically running air pressure to get the seed in the ground. That's where you have a lot more risk of that seed treatment kind of coming off the seed, getting up the air. And there's been some good advancements in reducing that. But that's kind of one of these areas where people maybe haven't dug into some of these new, or how some of these newer planters work. Some of the older ones, we're still looking at Telp or some other products just to kind of keep it moving smoothly through the planters, so you aren't getting all of that seed treatment coming off, and that way it stays in the ground instead of going in the air. And a similar topic, runoff bleaching. That one's pretty straightforward. I think we'll be talking a lot about that later, so I'll kind of leave that one be. But the other big one is for insecticide resistance management. We want to rotate insecticides, so we aren't using one insecticide repeatedly throughout the year or even across years. That builds up resistance quicker, so we kind of lose those tools pretty quickly. So this is one where if you use a seed treatment, well, if you go and spray a neonicotinoid foliar insecticide later in the year, you're really upping that risk of insecticide resistance. Then spills and disposal. I'm not sure if this will come up later, but a good example was down in Nebraska. Some folks were disposing of treated seed that they didn't need. I think it was going, or basically being disposed of at an ethanol plant, and you had contaminated ground where the concentrations of that seed treatment neonicotinoid was quite a bit higher than allowed. So those kind of situations, those are all things to avoid. That's basically all on the label or in these BMPs. So that's pretty much what I covered there. We talked about soybean aphid a little bit earlier with Hunter. I kind of wanted to mention one of the studies I've kind of used a lot when talking to farmers. This Christian Krupke was mentioned before too. He's done a lot of work, but this is work from a lot of folks across the Midwest here. So you might recognize a couple. I know Bruce Potter used to get out this way when he was still working. He's retired now. Aaron Hodgson from in Iowa, Jane Corporal at MDSU. But these two graphs is what I pulled out of that one study. This is something I show, basically showing what's the return on investment for these seed treatments for soybean aphid. This was back in 2013, so you think, we had pretty good crop prices. This is also a heavy aphid year. Over on the left, this is when we use scouting. So if you hear about the 250 aphid threshold, that's where we have folks out there looking to see, okay, do you have enough aphids that's worth using a foliar insecticide for? So not the seed treatments. And in this case, I should first say that vertical line, that's showing where 0 is. So you have a pretty good chance of getting a net return per acre using that scouting method without worrying about seed treatments. And in some cases, you're getting a pretty high return on investment, actually. That's in hectares there, so just convert that over, that's about 2.5 acres per hectare. Go over on the right, though. This is with a seed treatment, and there's not as good of a chance of getting a return on investment. In this case here, you might be losing a bit of money using those seed treatments. Some of the farmers in some of these scenarios actually were getting a bit of a return on investment. Usually seed treatments, we'd say like $5 an acre, seems cheap. So technically in the positive, but not by much, but you're also gambling quite a bit here. So this is one where I'd say your better chance for return on investment is using the scouting, the seed treatments, like we're mentioning, just not great efficacy, or usually they're too late, or rather the aphids show up too late in the season for those seed treatments wear off. All right. So what do you do as a farmer when you're thinking about, okay, well, what is my risk for the next year? Because you do have to do a little planning ahead of time. We usually don't want to be applying insecticides just on a schedule. That's kind of that prophylactic term, if you haven't heard of that before. Usually trying to avoid that. One thing I do a lot is actually pest forecasting, so we can actually use winter temperatures to get an idea of, are soybean aphids, for example, going to survive the winter? And in this case, it's close to -30 about is when things start to really happen in these maps. This is just showing winter lows. I think I had it for 2023, because that was, or 25 rather. We had it a little bit colder, not too much, I'd say though. But here's how that map turns out when we look at mortality for soybean aphid. In this case, we're getting, maybe about 10-20% mortality across central Minnesota. Higher in the red there, so that's getting into 75-90% mortality. So for southern Minnesota, you might get a little higher risk, especially if you had a high aphid populations the previous year. So that might be one where farmers might be wondering, okay, what do I need to manage for this year? Versus if it's an even colder year, have a good polar vortex, maybe you don't need to be as worried about soybean aphid or some of these others. So that's kind of what, kind of that question about what do we look at for risk in those individual fields. Farmers do have to make those decisions still. And it's not always one where we're out in the fields, making decisions for the next year all the time. Sometimes there's a little preemptive work going on beforehand. And that's what he's questioning about the seed treatments. So what makes sense from the fields, I kind of mentioned before, these high level averages, basically showing that, yeah, a lot of these pests, especially our major pests, not really working too well. They're not always going to pick up on ones like this. I have seed core maggot feeding on soybeans, so that one can hit both soybeans. Again, it's pretty rare. If you're that farmer, though, who does have a major issue, let's say you have a high organic matter, you're using manure, maybe green manure on the cover crop side, those are all things that can attract them in. And if you actually do get that high risk scenario where you're getting heavy populations, that might benefit in that case. But for most farmers, if they're coming to me asking, should I use a seed treatment? It's pretty much going to be like you're not in that high risk scenario for the most part. So I wouldn't be using it widespread across farms. So that's kind of what we're trying to navigate a little bit in the extension recommendations. Looking at these rare pests, and they're rare in terms of they're rarely going to be an economic issue, but want to make sure we're not kind of leaving those folks without options there too. Because seed core maggot, there aren't any rescue treatments otherwise. You can try to manage it with changing the planting dates, trying to match up when that pest emerges. That gets tricky too, but it's a pretty limited toolbox for when someone does have an issue with that. So that's where we kind of rely more on field history and farmer knowledge. And that's kind of how we balance that a little bit, where we're trying to figure out how do we deal with this kind of larger topic of neonicotinoid use, where maybe there isn't good return on investment, it's being overused, versus those cases where we look at that. All right, last thing I want to kind of plug a little bit for the panel coming up a little bit later. How do we go about finding untreated seed? So at least on our farm, like I mentioned, like our soybean, we really don't have any problem getting untreated seed. Especially as an entomologist going through all these things, I can know that, yeah, I'm not worried about that risk for the most part, and I'm going to be out there scouting anyways. What do you do for some of these folks that, let's talk about seed core maggot. On our farm, I've never seen it in high numbers. We have a lot of high risk scenarios for that though, for contributing to that. So in our case, yeah, we still do have corn seed treatments going on, but that's also partly because it's harder to get hold of untreated corn seed, at least in our area. But on that soybean side, basically, if we're getting a hold of our co-op or seed dealers, especially in November, no issue there. But even if we've had some times where we had to get some extra seed and we call folks up, and I won't mention specific companies, but it's bigger seed companies, no issue getting untreated soybean there. So that's kind of the dynamic we're working with a little bit here. And that's what we'll hopefully cover a little bit more in that panel. So key thing there, I'll just say for now, is if you're talking to your seed dealers, ask early. That's something too where you can assess your risk and figure out that, yeah, you probably don't have to be worrying about some of these pests, and also hopefully spending a little less, especially as we talk about that in the current markets in the ag world for inputs. All right, I can leave a question thing up, and I think we're starting the questions for the others coming up here too.
Thank you. So this time, I just want to welcome back Shawn and Hunter. And we have about 10 minutes, so if you have questions for either any of these three speakers, now is a great time to ask. And Elaine, if you are getting online questions, we'll do our best to answer those. Here.
So when I started using this treated soybean seed, it was because of cyst nematodes. Is that still a problem or?
I can answer, let me answer this.
Just repeat.
Sure. The question was related to using seed treatments for soybean cyst nematode. I mean, there are seed treatments labeled for soybean cyst nematode, however, neonicotinoids are not labeled for control of soybean cyst nematode. So you'd have to use something like Ilevo has that on the package. There are other southern United States, Victa, which we don't have up here. But we don't really have a ton of seed treatments that are super efficacious against soybean cyst. And we're still looking at genetic resistance going from Piraino to 788 to something like PK, rotating that around. There's going to be some GM coming out of the pipeline, I think in 28 or 29 genetically modified versions, and industries are. But I guess the point I would just want to say is neonicotinoids aren't labeled for control of SCN.
Can chime in a little
Yeah.
extra too. Yeah, so soybean cyst nematode for some folks that maybe aren't familiar, that's basically a worm or a nematode. It's not an insect. So that's partly why the seed treatments aren't necessarily labeled for it. Not much efficacy in that. But I will mention like some
A little louder.
mentioning.
So sorry, a little louder, closer
Okay.
again.
All right, and mention this or get microphone here. So short version is nematodes, they're worms, not insects. Insecticides aren't necessarily going to work on this. But when it comes to some of these seed treatments for soybean cyst nematode, those are the ones that we're not recommending those across the board or anything. But if I'm weighing kind of a list of insecticidal seed treatment, nematodes, fungicides, those treatments for nematodes, those do get recommended a little bit more again in various situations, but definitely kind of ranking a little higher than the percentage.
Yeah. Yep.
Question in the back. Or I guess up front actually.
I had a question about the farmer survey when you were sending out that initial request, and you said, if I heard it right, previous seed treatments was a possible indicator of damage from pests that could be addressed through neonics. Can you explain why that is? Is that just if they've used it before, there's a higher likelihood that they had those pests? Or...
Well, sorry, that would actually be, it would be lower risk.
Oh, right.
Yeah, so yeah, just risk level.
Gotcha.
Sorry, the mic.
Everybody listen, we got a camera up there.
I think as Dr. Hanson said, higher organic matter soils tend to have a greater need for treatments. What do you consider higher organic?
Can you
Yeah, so-
repeat the question?
Yeah, the question was about high organic matter soil being a higher risk for some of these soil-borne pests. And I don't have like a clear quantified number on that exactly. That's kind of the recommendations we have where it's more, is there, you might have soil tests where you're getting a rating of organic matter. I'm thinking more about just organic material out there that draws in insects like seed corn maggot. So that's more of kind of a categorical thing where, if you have a lot of material out there that might attract them, and that's what we're thinking about there for that issue.
So if you have a lot of crop residue from the previous year.
That can, sometimes it's more green residue or recently, I think Hunter, you could probably comment more on some of that maybe.
Yeah, I mean, from what I've read, yeah, like that's why cover crops, so you have that green organic matter or manure, that kind of stuff.
That does remind me of something. Kind of a follow up there. This is not necessarily just organic matter, but one I've been working on a lot is true armyworm that does get attracted in the cover crop situations if you don't terminate that early enough. So this is one of the challenges we're navigating. Well, sometimes seed treatments, neonic seed treatments are recommended for true armyworm. But if you have high numbers of that, they can kind of overwhelm that. So there's some other methods that are being used. So that's kind of in the framework a little bit, but one of the challenges the cover crop world we're dealing with is some of the cover crops can draw in some pests as well. And that's the attraction to sometimes organic matter is kind of wrapped up.
Are there,
Let's go in the back and then one over the wall next.
are there any specific cover crops that are problematic with bringing pests into the?
So the question was on what are there any specific cover crops that could be problematic for attractiveness? And the big one has been cereal rye, at least this true armyworm issue. Any grass they're attracted to in that case. But otherwise, you know, for a lot of these other insects, wireworms and others, Hunter, I'll pass that off to you. But in that case, it's I think just any green material, isn't that for the most part?
Yeah, they like grass, so like pasture, that kind of stuff. I think like especially white grubs they're a big pest in golf courses and stuff like that. So that was another high risk thing, was coming out of pasture. There's a question.
Somebody comment on that. I mean, the other thing to think about cover crops is, at least for us in the northern environment, depending on what your end use of this, if you want to capture phosphorus, you want to capture nitrogen, you want to use it as a weed control, I mean, that's going to dictate when you terminate it. And for us in Wisconsin, our threshold was roughly, gotta pick a number, magic number, 4,500 pounds cerealized biomass is what is needed to reduce your water hemp population by 50%. Okay. So again, 4,500 pounds of cereal rye is basically this tall, 36 inches. So if you're going to, that means you'd be no-tilling into that. That's where we get into these challenges with slugs and other insects when we use the neonicotinoid in that situation. And if you're terminating, because we used to tell farmers to terminate 2 weeks before planting, gets ready with this green bridge, we don't have that. Well, 2 weeks before planting in Minnesota, when you get like 4 pounds of biomass out there, you know, it's not that low, but you know what I mean? It's not a ton. So if you want to be effective, I guess, just depends on what you're using your cover crop for, what's the purpose of it. That makes sense, everybody.
Let's get the question over there.
Yeah, I believe that you said that the seed treatments are applied at the factory for corn and at the co-op for soybeans. And I'm just wondering why that is. Can you tell a little bit more about that?
Yeah. I mean, there's the seed industry reps here. They can probably do a better job explaining it to me than I'll be talking later this afternoon. But generally, corn is applied, you know, before it leaves the facility. Soybean is usually treated downstream. That's an added benefit to the co-ops to be able to apply that service to local farmers and make these choices downstream. That's just basically how seed is handled. Difference between treating corn seed and soybean seed is treated within the seed industry. So I guess a wrong seed industry can correct me. Perfectly fine. He's corrected. Yeah, I am married. So, trying to think. So that's kind of how we do generally, but you know, I'm not going to get into all the nuances here, but a lot of time that's a benefit for a co-op to put in a treater. They can charge a farmer like $5 an acre to treat seed. It's kind of added revenue at that co-op. So there's some other nuances that are involved in that, and the rates, timings, the amount of seed. There's a lot of different nuances there. It's not the application technology on corn versus soybeans, like slurries, whatever. We talk about it just for an hour. So in short, that's usually what happens. Yeah.
Are there any naturally known predators that could keep the insects at bay? I know you mentioned it a little bit of one of your research projects that you hope to do.
Yeah, I was kind of speaking generally.
Please repeat the question.
Oh yeah, sorry. The question was, are there any specific natural predators that could keep these pest insects at bay? Yeah, I was kind of speaking generally. I think there's a lot. I mean, I know Bob's lab, there are a lot of parasitoid wasps. That's kind of a big area of biological control. So those are the tiny little wasps that sometimes microscopic. They lay their eggs inside of pests. The eggs hatch and then burst out, kind of like the movie Alien, and it's very effective. So that's been a big area of biocontrol and actually discovering new species, even also in Bob's lab. So really exciting. Yeah, I mean, like ladybugs are obviously a huge predator of aphids. I know there's predatory nematodes. But yeah, since my focus, I'm a master's student, so I really have to like kind of narrow in, and I'm just kind of looking at neonicotinoids and stuff like that, and I sort of of this other stuff. So yeah, that's really interesting. Yeah. Nothing else to add.
Okay, we've got one more question and then we're gonna move on.
Good question.
What role does integrated pest management play in purchase or sale of neonicotinoids?
We'll get an open-ended question, but kind of a big one. So the question was, what role does IPM or integrated pest management play in the purchase of seed treatments, neonicotinoids? And I guess I could say I wish there was more of a role, but that's kind of this question of how do we get ahead of kind of farmer perception of risk? And that's one where, if they're dealing with all these things like Shawn mentioned, just kind of the either insurance or pre-plant decisions, that's maybe not the IPM side, but really it's, do you need this and are you getting return on investment? And I think that's one to kind of get word out more on. It's, I think, what may need to happen there, but I think there are some folks like we've seen where they do manage, at least on the soybean side, where they don't get those treatments because they know they don't really need it in that case for the most part. So I think it just varies depending on adoption, this question. So it's out there, but there probably could be more drifting towards focusing on IPM on that side. You have time for one more question or?
Sure, yeah.
Insects, for every one bad one you kill, you're killing 700 plus good ones. So, as we fight one, we focus on one thing, kind of like the cover crop. That's just part of the whole system. And if we just really look at cover crops alone, you can have problems. You look at it as a whole system. I haven't had armyworms for 10 years. Of course, now that I say that, but it's a whole system. You can't look at it just individually, like, oh, we're going to attack this problem and get blindsided by something else.
So I guess the question was more kind of on system-wide things where we're targeting one insect, we're often killing a lot of other ones. I guess I'll add on to that too is a lot of our insecticides like Neonics are broad spectrum. That does harm some of those natural enemies out there. Versus some other insecticides might be a little more targeted, or you get to like your BT examples. Those usually tend to not bother the natural enemies. So yeah, kind of those choices play a lot there. Katie, you had your hand up in the back. We have time for one more question. We gotta cut it off. We can chat later anyways back there.
Sure, yeah, just go ahead. Then we have to go.
I was just wondering, there was a work done that actually showed the residues present in roots, stems, leaf material and all that. Basically, has that been done? I think that was done on what? That was soybeans?
That was the question was related to the residue work that was done on soybeans. Yes.
Has anything been done on corn or any of the other major things that are treated?
And not that I know on corn. I mean, Dr. Christian probably is the one that's doing that soybean right now, and he was kind of surprised it hadn't been done. I'm not going to guess what the industry has. Not even going to guess. It's not my place. But in the public arena, this is the first I've ever seen of anything like this being published. How much is available in these different plant plants?
Yeah, it's very. What is the only difference be dependent on the vascular system of the plant, and that wouldn't be much different?
The question was, would that only be different based on the vascular difference of that plant? And then his response was that probably wouldn't be much difference, and now it's probably. And it's basically where it's going to go. You just have to understand the difference between a soybean seed, because it's cotyledons come up and time's up.
Thank you.